Hi, everyone. Welcome. Welcome back. Feels so good to be back. Yeah. This is the inaugural 2021 Coffee and Conversation with our guest, Burt Adelman, and our other guest, Will Thorndike. And yeah, I want to welcome you all to the event and also to the first ever event in the Davis Center for Human Ecology. So you are the first. And therefore I need to take a photograph. Yeah. All right, sorry, Bert.
Oh, you want me to get out of the way?
No, you're good, you're good. Okay, everyone wave. Good. No, honestly, at 7:30 this morning, there was an enormous roll-off dumpster in the back, and E.L. Shea was here dealing with a beeping fire alarm. We are still a work in progress, but we're very, very excited to be able to use the building. There are bathrooms right to the left here. The only rules are do not walk on my hydro seed as you leave or exit the building. That will be really frowned upon, right? No, I'm teasing. No, I'm not really teasing that much. But so today we're going to have a discussion. You can see someone's already written on our whiteboards, the inaugural use of our whiteboards, which is great.
They look very clean.
They look clean. They look clean.
I was a little nervous.
And my role today is only to welcome and then to introduce Will Thorndike, who is our interlocutor this morning. Will founded Housatonic Partners, a private equity firm in 1934, although he looks a lot younger than that. No, 1994. I've known him for 10 years, and across those 10 years, he's been an incredible mentor to me as a member of our board, chairman for 5 years. Beyond his expertise and knowledge in private equity, he's an amazing storyteller and writer, and he's written on topics ranging from Ben Franklin to high school football to obviously business leadership, and just really recommend that you check out some of Will's writing. He has incredible range, like we like to say. He's rangy, and that ranginess is exactly the kind of thing we like to inspire in our students here at the College of the Atlantic, and we're gonna get a little bit of that today, I think. So without further ado, I'm going to introduce Will to begin the conversation with Bert. Thank you all.
Thank you, Darren, for that gracious, overly gracious introduction. So thank you. I'm delighted to be up here with Burt Adelman, the co-founder and chairman of the board of Verve Therapeutics. Burt, in addition to being a longtime friend of the college, is an internationally recognized expert in the field of drug development with more than 25 years of experience in the biotech industry. He was most recently executive vice president and chief medical officer at Dyax Corporation, where he led the development of Takhzyro, probably not perfectly pronounced, which was approved by the FDA and EMA for the prophylactic treatment of hereditary angioedema. Dyax was acquired by Shire Pharmaceutical in 2016. Dr. Edelman began his career at Biogen, where he had an exceptional track record, having led the successful development of major biologic therapeutics, including Angiomax, Avonex, Emaviv, and Tysabri. At Biogen, he served in various roles, including executive vice president of R&D and executive vice president of portfolio strategy. Dr. Edelman is a member of the board of directors of Catabasis, Sirona, and Clear Creek Bio. He is also a special advisor to Nova Ventures, a leading biotech venture capital firm. He graduated from Cornell University Medical College, served as house officer and medical resident at the Peter Bent Brigham Hospital, now Brigham and Women's, and at Harvard Medical School. Dr. Edelman is a member of the Tufts Medical School Board of Overseers, an advisor to the Boston Biomedical Innovation Center, and a member of the investment committee of the Partners Innovation Fund, which is a collaboration between MGH and Brigham and Women's. Bert, it's wonderful to have you here this morning. Thanks for joining us. I think rarely in the history of Coffee and Conversation has there been such a gap in scientific background as that between you and me. So let's dive in and maybe start with some definitions. And we're going to get to the whiteboard in a minute. You'll see that Bert has populated the whiteboard, as Darren said. But maybe start with what's a vaccine? Project more? Okay. So we're going to start with some definitions. What's a vaccine, Bert? How would you define that?
A vaccine is a kind of drug, and by real introduction, I like to think of drugs as toxins that have reasonable good attributes that we take advantage of. A vaccine is usually a product composed of biologic components as opposed to small molecules. So, aspirin is a small molecule, an antibody or milk albumin, they're all proteins. So a vaccine is a therapeutic strategy to trick the body into making antibodies and other immune responses to a foreign substance that the vaccine introduces that will enable us to be protected from a viral infection, a bacterial infection, or even the effect of a toxin. So, for instance, a rabies vaccine is really not intended to keep us from being infected by the rabies virus, but also to protect us from the toxic effects of that virus. Tetanus toxoid, even a better example. And obviously, in the case of COVID, the purpose of the COVID vaccine is to protect us as individuals from infection by the virus. But just as importantly, and I think what's become, unfortunately, the political discussion around COVID vaccine is the intent of vaccination for contagious infectious illness is not only to protect us as individuals from infection, but to secondarily, and just as importantly, provide protection for the community, for the population of all of us from the infectious agent. And that requires a vaccination program in addition to vaccine. And as we all know, the vaccination program, very simply put, is the undertaking to get as many people in the at-risk population vaccinated as possible. And when I say at risk, I don't mean because you may have medical issues that put you at greater risk than someone else for a bad outcome. But I mean the at-risk population in that we all live together in a community where the virus is present. And to the extent that we allow the virus to continue to be present, we all are at potential risk of adverse outcomes from an individual viral infection.
Great. So maybe a place to start is, how did we get here? What allowed for the extraordinarily rapid development of the COVID vaccines? And I'm sure as you go through that, you may tick through some of the items on the whiteboard. But
So great question. The reality is, of course, although COVID-19 was a new virus that we had never seen in the history of studying viruses, coronaviruses, of which the COVID virus is a member of the family, are very well-known pathogenic viruses. On the sort of least concerning level, many simple upper respiratory tract infections, bad cold, maybe flu-like symptoms, can result from coronavirus infection. And then, as many of you may recall, we've experienced two very serious coronavirus infections. One, the first being SARS, which was, I don't know, maybe 10 or 12 years ago, which came and went very quickly. But unfortunately, for those individuals who got SARS, severe, acute respiratory syndrome, the outcome was usually death, or maybe a highly morbid outcome and lung transplant. But that came and went pretty quickly. And then the second one was MERS, which Middle East respiratory syndrome that we think may have emerged from camels. And that was also a very serious, highly morbid infection that came and pretty much went, although I'm not actually a virologist at all, came and went and has come back a little bit. But, and in part, paradoxically, those viruses came and went so quickly because they were so lethal. They didn't have the opportunity to sort of spread around amongst the population, as opposed to, say, the flu or the COVID virus that we're dealing with. However, scientists have been studying these viruses for a long, long time, and certainly SARS and MERS really got people focused on coronavirus and trying to understand the critical factors of coronavirus that enable it to infect host cells, basically the upper respiratory tract of man, and how you might fight or prevent that from happening. So that when the SARS COVID-19 virus emerged, there was actually a lot of background knowledge about this class of virus. And it was, in a good way, a perfect storm of scientific and medical capability to focus on this virus very quickly. Fortunately, let's leave politics and various, commentary aside. In reality, the seriousness of this virus was recognized very quickly. The ability to bring highly sophisticated, highly accurate, and fortunately, relatively rapid molecular biologic and protein biologic technology to bear led to a very rapid sequencing of the spike protein, which I didn't put up there. But, simply put, people already, before this happened, people already understood a lot about the mechanism by which SARS virus enters human cells. And they knew that it was a lock and key strategy where this protein on the surface of the coronavirus, called the spike protein, because it looks like little spikes all over. You've seen these images a million times. We knew that that spike protein found its way to connect to a protein on the surface of human respiratory tract cells, and used that sort of like Velcro, used that that attachment to actually invade the human cells. And all viruses have to get inside our cells to do their nasty businesses, because viruses are, a philosophical discussion could be had as to whether viruses are alive or dead. In the environment, they are, at a minimum, they are essentially inert. They can't reproduce. Bacteria can reproduce on minimal nutrients, but a virus does not have inside itself the equipment to actually reproduce. So viruses have to infect a host cell, be it human or bacteria or an animal or an insect. They have to infect those cells. They have to get inside that cell, and they have to take advantage of reproductive machinery, enzymes, the ability to make DNA or RNA, depending upon what kind of virus it is, that is inside our cells for them to be able to reproduce. And then they reproduce within the cell at such great numbers that they essentially lyse the cell and they destroy it. And the symptoms that we experience from a viral infection are the combination of the effects of these cells being destroyed. So in the case of flu, a cold, or SARS virus, they're very destructive to the cells of the respiratory tract. And remember, those cells have all these wonderful cilia on them that move gunk and mucus up and keep our lungs clean. And that when these cells die, we can't manage the secretions that are within our cells, and they simply, within our lung, and they simply accumulate in there. And then it becomes like a petri dish for bacteria and other things to happen. And obviously, they also interrupt the exchange of gases in the little cells in the alveoli. But basically, we feel sick when we have a viral infection, one, because the normal function of the affected tissue is disordered, and because the body initiates an immune response. And the immune response is basically a good thing, and it's intended to do two things: to prevent the virus from entering more cells and to actually kill cells that are infected by the virus. In the case of SARS, one of the reasons, or the predominant reason we think why people get very sick, often end up in an intensive care unit, even on a respirator, is as damaging as the infection is to the intrinsic cells of our lungs, the immune response to the infection is excessively exuberant, to the point where our own lung tissue becomes the innocent bystander of this immune response. And you now have infection and this sort of runaway immune response, sort of like, autoimmune disease, lupus, rheumatoid arthritis. These are immune diseases. We don't think they're related to virus, but they might be, where the body makes antibodies or activates T cells that damage our own tissue. Very similar thing happens with SARS. Fortunately, not quickly enough, but we quickly learned that if you give people steroids, Decadron, for example, you can tamp down that immune response very significantly. And the very early on improvements in survival were simply due to recognizing that steroids work. And you might say, well, why didn't we just give it to everybody to begin with? And the answer is because people have been studying steroids in acute lung injury for years, and 99.9% of the time they don't work. And fortunately, in the case of SARS, the subtlety of the immune response was such that it responds quite reliably and predictably to an intense course of steroids. The steroid that people tend to use is Decadron. So that's why people get so sick when they get SARS. So the good news is, back to spike protein and all of the biological capabilities, very shortly after this infection was recognized as being a very dangerous and rapidly spreading viral infection, we knew to look at the spike protein, and the sequence of the spike protein, that is the amino acids that are in sequence to make the spike protein, which are how all proteins are made. They have a series of amino acids, was published, and it became part of the everybody could see it literature. And therefore immediately companies that were interested in vaccines, in making vaccines with various different technologies, were able to identify that the spike protein was likely a really good target for vaccine development, because if you could get the body to make an antibody to interfere with this, lock and key or Velcro relationship, if you could grease, the little hooky part of the Velcro so that it wouldn't hold on, you might be able to prevent viral attachment. And if you prevent viral attachment, you would either eliminate or very significantly tamp down the infection. So a number of companies took that information immediately and put it into the vaccine development program that they had already built in their company. And in the case of two companies, one called BioNTech and the other called Moderna, these are companies that had been examining the use of mRNA, messenger RNA, as the fundamental component of vaccine, of a vaccine that they would could give to people. And as exotic as this may sound, it's actually taking advantage of age-old understood cell biology. So we all know we have DNA, and DNA contains sequences of nucleic acids that define the code for making a protein. So they hold the information that says which amino acids need to be hooked together by the cell in what order to make protein, whether it's spike protein or keratin that makes our hair, or proteins for our skin, or muscle protein. All that information is wound up in our DNA. For a cell to make protein, the first thing that has to happen is the DNA has to be opened, and then it has to be read, because the information that's in the DNA, which is in the nucleus, has to be sent into the cytoplasm of the cell, which is the machine industry part of the cell. So the nucleus is sort of like the storage, the data, the hard drive or whatever, of all the information. And the cell is sort of where actually the business of life, the cytoplasm, the business of life is carried on. So what happens, and is the DNA opens and it's read, and that information is translated into messenger RNA, which is essentially a mirror image. And that messenger RNA is transported out of the nucleus and into the cell, where there's a system that actually reads the messenger RNA and takes that information and makes a protein, puts all the amino acids together. And lots of Nobel Prizes have been given for that, and it's routine stuff, happens every day. So Moderna and BioNTech were companies that were interested in using mRNA as the basis of a vaccine. And the idea, very simple. If we can make a piece of mRNA that will tell a cell how to make the spike protein. It's got all the information there. And all you need to do is put that mRNA into the cytoplasm of a cell, and the cell, they're not that smart. They're like, oh, here's a message, I got to make a protein. No, they don't ask, is that the protein I really ought to make? It's like, here it is, it's a message, I'm going to read it, and I'm going to make a protein. So the idea is basically, if we can get message RNA that reads for the spike protein into human cells, the cells will probably make this protein. It's never an absolute. They'll probably make this protein. They'll display it on their surface, or maybe excrete it. And the body's immune system will say, what's this stuff? It doesn't belong here. And it will be turned on to make an immune response. And the immune response will be antibodies that bind to that spike protein. They're like little fighter pilots. They'll find it. They'll be in the bloodstream. They'll be in the extracellular space, all the liquid between our cells. They'll be everywhere, because when we make antibodies, we make lots of it, and it goes everywhere. And they live for a long time, and we keep sort of making them. Plus, the other half of the immune system will also be turned on, and special cells called T cells will be activated, and their job is to sort of find human cells that are infected with the virus and clean them up, so the virus can't continue to multiply and spread. The challenge is, for BioNTech and Moderna, the challenge is it's never really been done before. Been done in animal models, but never in humans. mRNA is hard to make, very expensive to make, and very unstable. You look at it the wrong way and psh, it's gone. And there are enzymes everywhere in the world on our fingertips called RNase, and then they just chew this stuff up. So it was a bold suggestion that they could make all this happen, and they could make enough RNA, keep it stable, and then they had to deliver it. So because, as I just said to you, mRNA is so unstable, so fragile, and our body is full of enzymes to chew it up because, you know, we really don't want foreign mRNA getting into our system and sort of taking over the show. So it makes sense that we don't want to be able to make a happy home for mRNA that's injected into us. So they had to come up with a way to package it whereby it would remain stable and could be administered through injection to people, and then get to some place where the mRNA would be introduced into cells that would then make the spike protein, and it would last long enough for our body to say, you know, holy cow, what's this stuff doing here? We got to get rid of it and mount an immune response. Because if it turned out that the spike protein had a really, really short half-life in our bodies, it wouldn't be there long enough for an immune response to happen. And nobody knew the answer to that. So there were a lot of gaps in knowledge, but they figured out that they could make the mRNA and they could put it in something called a lipid nanoparticle, a tiny little microparticle, much smaller even than a cell, made of various lipids, actually a number of lipids, simple lipids like cholesterol and a few others. But there's one very special lipid in these things. It's a charged lipid. So it has charge. And it's critical because the way the lipid nanoparticle is manufactured, until I got into this field, I used to think, you know, naively, that you make these lipid nanoparticles and then you do some magic and you inject the mRNA in there. But in fact, what happens is you have to mix all of the components together. And the mRNA is a highly charged molecule, and this charged lipid is of opposite charge. So they find each other, and then that sort of seeds the process, and this lipid nanoparticle then forms around the mRNA cargo. And then it's that lipid nanoparticle with the mRNA inside it, which is the drug or the vaccine that, you know, I mean, I don't know, how many in the room got an mRNA vaccine? I got Pfizer, and everybody else either got Pfizer or Moderna. Anybody get J&J? Yeah, so J&J is an excellent vaccine, despite the little issue that happened. It's a different technology. So it's a virus technology. If we get a chance, we can talk about it. So, BioNTech, you know, did a deal with Pfizer. Moderna didn't have to do a deal with anybody. They had tons of money. You know, paradoxically, Moderna as a company was formed to deliver mRNA to the liver repeatedly to try to solve problems like hemophilia, where people couldn't make a protein and therefore had, like in the case of hemophilia, a bleeding disorder. The problem they ran into was that first of all, mRNA has a short half-life. When you inject mRNA into somebody, you make some protein, it works for a while, and then the mRNA is gone, which is a good thing. The problem that Moderna ran into is that repeated administration, at least of first and second generation lipid nanoparticles that were going into the liver, turned out to be toxic, and they couldn't do it. Now, the world has changed. That was a number of years ago, and they switched into vaccines because vaccines are basically once and done. Lipid nanoparticle biology has advanced significantly, and they're sort of back working at some of the old stuff they were doing, and we at Verve, which we may get a chance to talk about, do a lot of that. So it was a bold assertion that this would all work, but it was also, a compelling argument based upon a lot of biologic understanding. And not to trivialize it, but at the end of the day, it's really, we've been injecting mRNA into cells in culture and into animals for years. And we know that when you get it in there, it's going to do its job. The issue is, can you get enough? Is it going to last long enough? And is the spike protein going to be expressed in a way that the antibodies that are made against it will work against the virus? All of that, complete unknown. But, we didn't have a lot of options. And again, the underlying assumptions are compelling and supported by, lots and lots of science. So they went ahead and figured out how to make lots and lots of mRNA that was of high quality and keep it stable. And they figured out how to make a lipid nanoparticle containing mRNA. I might add that charged lipid that makes the whole thing happen, that is a patented product. So very valuable to the people who own the patent, and very hard to manufacture. And when you hear about early on about the manufacturing shortages associated with making enough of the Moderna and or Pfizer vaccine, it was because the availability of these charged lipids was very limited, like single source kind of suppliers. The lipids are different. Moderna has their own LNP. Pfizer has a lipid LNP that was developed by a tiny little company in British Columbia that I know because we have a deal with them also, who had never done an experiment even in a non-human primate with that lipid nanoparticle when BioNTech took it from them.
So as you go through that, Bert, two questions.
Yeah.
If COVID had arisen 5 or 10 years ago, how much longer might it have taken to develop a vaccine? And then secondly, as you described that, some of the factors that allowed for rapid development were specific to COVID, and some were more generalizable. So if you view it as a broader technology, will it allow for more rapid, lower cost development of vaccines for other diseases going forward? So those are two questions.
Yeah.
First one's
So, on the one hand, it's a really scary question, which is, like if this happened 10 years ago and Moderna didn't even exist, BioNTech didn't exist, the idea of mRNA as a vaccine was like maybe somebody's dream. The good news is another excellent vaccine is the Novovax vaccine, which will probably be approved shortly. It wasn't approved earlier in part because they got started later and because they it was harder to run the trials as more and more things were going on. But the Novovax vaccine, which I think has almost the equivalent efficacy to the Moderna and Pfizer vaccine, is actually a very conventional vaccine. So what they do is they express the spike protein in culture. So, they have big tanks that grow protein cells, and those cells make the spike protein. And then they mix it with something called an adjuvant, which we've been using forever in vaccines. So maybe if you've ever gone in and gotten a vaccine and noticed the doctor shake or the nurse shake it up before they give it to you, it's not clear. It looks like gunk. And you're like, do I really want that gunk injected into me? But that's the adjuvant. So think of the adjuvant as material that irritates your immune system, literally, which is what it does. It irritates your immune system. It's why your arm hurts when you get a flu vaccine. It irritates your immune system, sort of turns it on, and accelerates the immune response. So the Novovax vaccine, very traditional vaccine technology.
So 10 years ago, that would have been
Yeah.
the
Now,
solution.
however, 10 years ago, we also would have tried to make attenuated virus vaccines or dead virus vaccines. And in the case of SARS, the dead virus vaccines, and I think CureVac just released some results of a dead virus vaccine, which is also mixed with adjuvant, doesn't work very well. So the good news is one of the conventional technologies did work. And of course, the Novovax vaccine, easier to store. You know, it's as everybody probably knows, if you were in the deep freeze business, you probably ended up making more money than Pfizer did, because the Pfizer vaccine needs to be stored at -70. The Moderna vaccine, also a lipid nanoparticle, we don't know, because it's all proprietary. We don't know whether it's because their lipid nanoparticle is a little more robust than the Pfizer one, or it's just a timing thing, and Pfizer didn't spend enough time trying to perfect the environment in which they freeze the vaccine. But the Moderna vaccine is a little more robust, doesn't need to be stored at -70, actually has a certain amount of shelf life at regular refrigeration. The Novavax vaccine probably could be left out in the sun.
Interesting. So I want to move on to
Yeah.
Verve and a couple of,
Yeah.
so in terms of the generalizability, the applicability going forward of mRNA for other diseases, high,
I
medium,
think
or low?
generically speaking, absolutely. The question will always be, when you give people the mRNA for whatever target you've got in the virus, is that protein going to be expressed in a high enough concentration for a long enough period of time and in the right three-dimensional configuration, so that when the body makes an antibody to it, it will find the target on the virus and do whatever it's supposed to do. So this is not, there's no blank check to be written here that, yeah, we've solved the problem and we're ready to go the next time a pandemic virus comes. We may be, we just don't know.
All right, so let's turn to Verve, and
Sure.
I guess the problem you guys are trying to solve and how you're trying to solve it.
Sure. So Verve is a company that was very fortunate to be invited by unbelievably bright people to be a part of and a co-founder. Not because they thought I was particularly intellectually adept, but because he's pretty good at keeping the trains running on time. And somebody in this group of brainy Harvard scientists has got to keep the trains running on time. And that's how I got involved. So this is a cardiovascular disease company that is focused on eliminating permanently atherosclerotic cardiovascular disease, which is still the #1 killer in the world. So, I mean, this is a incredibly bold, way out there kind of idea. But actually, it's an idea that I think, and in the 3 years that we've been doing it, increasingly so, is really not going to be potentially that hard to do. It's based upon some very fundamental understandings of why people get atherosclerotic cardiovascular disease from a genetic perspective and from a protein perspective that were in part discovered and evolved by one of my co-founders, a fellow named Sek Kathiresan, who's a genetic cardiologist who he still is affiliated with Mass General, but he's now our CEO, and a few other folks in this founding group who either worked in Sek's lab or were longtime collaborators. And what we now clearly understand is that atherosclerosis, hardening of the arteries, thickening the arteries that causes heart attack and stroke, and in some people can cause peripheral vascular disease, is the result of excess lipid accumulation in blood vessels and an inflammatory, destructive process that occurs when that happens. And we are all, to some degree, at risk of this. And the two most significant lipid constituents that drive this are LDL-C, which is a particle in the blood that contains lipid, and triglycerides. And LDL-C is the bad cholesterol and is probably the most significant player in this sort of pathologic dance. What Sake and his colleagues and some others have learned is that there are approximately 4 genes that regulate 4 different proteins that ultimately control LDL levels. And they're all in the liver. And, Sake has proven this and others in 2 ways. We know that individuals who hyperexpress, and they're rare, but they're around, people who hyperexpress or have an aberrant hyperactive form of one or more of these proteins have very accelerated atherosclerosis. Some of these people will have heart attacks in their late 20s and are lucky to live, through their 40s, no matter what they do. And if some of you can remember, like Jim Fix, the guy that got us all to run, who started running because I don't know what his family had, but they clearly had a very malignant hyperlipidemic predisposition because his father had died at a very young age. And remember, despite being a skinny, hard-running guy, he died in his late 40s or early 50s of atherosclerotic heart disease. So we know that people who overexpress one or more of these genes have a problem. And we know that people who are deficient, either totally or partially, in one or more of these genes, usually it's only one, even if they instead of work out, they lie down until the urge passes. And even if they, eat Ben and Jerry's every night, and even if they look a little plump, we have a picture of the woman that Sake met, who we discovered the first of these defects in. She could lose a little weight. They never get atherosclerotic heart disease. They just do not get it. And furthermore, we know that some of the drugs that are currently used, some of which target one or more of these proteins, are all effective in reducing heart disease. The problem is they're drugs. And as I said, drugs are toxins that have benefits and deficiencies. And we tend to not want to give them to people until they're on in life. And then people tend not to take them. Bill Clinton's the best example, right? Bill Clinton had very bad atherosclerotic cardiovascular disease. He was put on, a statin. He had apparently a very good response. So good that, like most normal people, he stopped taking it. He said, I'm fine. And then, as you know, he ended up having quadruple bypass surgery. So the problem with conventional therapy is, first of all, this is a problem that hundreds of millions of people have. People are very bad at taking their drugs, chronic drugs, routinely, and drugs have side effects. So, Sake and some of his colleagues proposed, let's basically come up with a once and done strategy to prevent atherosclerosis. If we can knock out one or more of these genes, then people's lipid levels will drop. We know that people who are born missing one or more of these genes live a normal, healthy life. So the risk of, you're playing with the genetic code and you knock out one of these genes, we think is not all that disturbing. And we also think that we can progress these experiments in man, beginning with people who have these highly morbid hereditary lipid disorders for which there is no adequate therapy, and then slowly move, as the circle gets bigger and bigger, we can move to larger and larger populations of people still at risk. And then maybe ultimately, many years down the road, maybe everybody would be treated when they were 12, and atherosclerotic cardiovascular disease would not be part of, the world of health as we know it. And not surprising, of course, the first venture fund that we were able to get to join this endeavor was GV, Google Ventures, who thought, wow, this is a really big idea. We're into really big ideas because we don't mind making money along the way, but we're into really big ideas. So 3 years ago, we founded this company and we decided to use CRISPR technology. That's a long story. That's
Number
how we're
2
getting
on the whiteboard.
So we decided to use CRISPR technology. And the reason why we decided to use CRISPR technology is because what CRISPR is basically a strategy that bacteria have. It's their immune system. And it was discovered by people studying actually over many years how bacteria become immune to viruses that infect bacteria. The world is full of what are called bacteriophage. They're just viruses that infect and very effectively kill bacteria. And bacteria have developed a system which is the derivative or the forerunner of what we call CRISPR to defend themselves. They figure out how to identify the virus, and they kill it, and they save pieces of its DNA, which they put into their own DNA, so that when the next time they get infected by that virus, they already know the right code to make a protective mechanism to chew up the virus's DNA. And I'm not going to go into huge details, but that sort of was what were the clustered regulatory interspersed palindromic repeats. That has to do with these repeats of DNA that were found in bacterial DNA that were the code for how to kill the viruses. The Nobel Prize was given last year in chemistry to Jennifer Doudna, of whom there's a wonderful book written, and Emmanuelle Charpentier. And they, along with colleagues of mine at the Broad Institute, who have been in a patent fight for years, and although the Broad thinks we won the Patent Institute, they got the Nobel Prize. So, we can figure it all out. So what they figured out was how to retool this bacterial system to work in mammalian cells, or maybe in insects, but to work in complex cells, and to use it to actually alter the DNA inside a cell. And altering the DNA is a very broad, non-specific term that could be translated into knocking a gene out, which we would call gene knockout. It can be used to insert a gene, which at the moment is pretty difficult. Knocking genes out, do that every day. Or further advances have been made at the Broad in particular by the winners of the patent, but the losers of the Nobel race, and they are upset, and I don't care. They figured out how to do one better than knocking a gene out or having to stick a whole gene in. They figured out how to do something called base editing, where you could either disable a gene by simply messing with the DNA code a little bit, or by actually fixing a gene. So you might be able to fix the gene because, for instance, sickle cell anemia, as terrible as disease it is a result of a single base pair alteration. So you could actually go into the hematologic stem cells and fix that one little alteration, and now people may no longer have sickle cell. And in fact, a company that we collaborate with, Beam, which is a base editing company, is in the midst of soon to start a clinical trial in sickle cell.
Wow. So I'm going to just,
Yeah.
I want to make sure we have enough time for Q&A.
Sure.
So That's an exceptional introduction to major complicated concepts, but at the forefront,
So let me just
driving all this. Yeah,
one
go
last
ahead. Go ahead.
So to sum it all up, how does this work at Verve? Well, we make an mRNA that reads for the enzyme, which came from the bacteria that cuts the DNA up. What Doudna and Emmanuel Charpentier got the Nobel Prize for, they figured out how to make this thing called guide RNA, which takes the enzyme to the exact right spot in the DNA to cut the exact gene at the exact space, to take advantage of the fact that when genes are cut, the cell is very sloppy in fixing them, that usually what happens is the gene is just lost. So in human application of CRISPR, we deliver mRNA for an enzyme that cuts DNA. We deliver a guide RNA that takes the enzyme to the exact spot. We put it all in an LNP, and fortunately for us at Verve, LNPs naturally go to the liver. So we can deliver this LNP very effectively at very low dose to liver cells to cut and therefore terminate one or more of the 4 genes of interest. And in fact, we already have a product for the first 2 genes on our list, and we hope to go into man with this in about 1.5 years.
So you guys are at the junction, the intersection of mRNA and CRISPR
Yes.
technologies, right?
Yeah.
Verve,
And
so-
we are probably, we and Intellia, some of you may have seen a company called Intellia announced the first human genome in the liver editing result, where they're treating a very serious rare disease, heart and nerve disease called TTR. And they demonstrated last week in a New England Journal article that they could selectively and relatively safely cut that gene in humans and demonstrate a remarkable reduction in the target that they're trying to eliminate.
Wow.
So this is real. It works.
All right, so let's open it up for broader Q&A. Chris, I will. There's a wealth of expertise to my left, including on the whole FDA approval process. There's a whole other set of topics there. But Chris, go ahead.
So I'm a biologist, so I'm going to ask a business question. So for these successful vaccines, it seems like there's 2 things you need. You need the high tech to be able to make the vaccine, but then there's these phase trials that have been really complex, and that seems to work independently of how good of a technique you have, and it's really been successful. So if you're a business like your business, to what extent do you become the expertise in phase trials versus doing what like Biotech did or whatever, like finding a partner to be able to do those trials? Because some of these vaccines that look like they might have been good just blew it in phase trials, right?
Yeah. Well, I mean, in AstraZeneca, which is a very big vaccine company, made some terrible mistakes. The challenge that everybody had, nobody knew what the right dose was. And the reason why we all got 2 doses of Pfizer or Moderna, it turns out that they were right. They were concerned that 1 dose would not deliver enough protein and that you would need a kicker down the road. And they knew from the hepatitis B vaccine, from the vaccines like Gardasil, the vaccines for papillomavirus, it was not unheard of that you need more than 1 shot. So they said, you know, no time to be wrong. The conservative approach is 2 vaccines. Now, you're talking about how do you get all these people and how do you get it all organized? It's very hard. And it's amazing they did it, but they had a lot of support. They you know, I mean, most people wanted to be in trials. Everybody wanted this to happen. You know, but it was still a miracle. It's a small miracle that they pulled it off. Absolutely.
Question
Yes.
over here.
Over the last several months, it became available to children over the age of 12. I'd like to understand how, in such a short amount of time, they then discovered it was okay to give it to children.
Yeah. So they were doing trials in kids. So that's a really good and complex question because children are like a protected class at the FDA. And on the one hand, the FDA demands that people like me, if I'm developing a drug that is relevant to children, they demand that I take the time and effort and expense to figure out if it's safe and effective in children. But usually they want to know that it's really safe and effective in adults, way ahead of letting me do trials in children. Here, I think the issue is that, well, on the one hand, a vaccine is a vaccine is a vaccine. So yes, there are some odd occurrences where vaccines turned out to be uniquely toxic in kids, but the vast majority of experiences is a vaccine is a vaccine is a vaccine. An immune response is an immune response is an immune response. The good news is kids tend to get better immune, younger people get better immune responses than old people. Maybe kids will only need one shot. But I think in many ways, again, I'm not a vaccine guy, but in many ways, I think the risk versus the benefit here is in favor of accelerating the use of these vaccines in kids. And I think some people can make an argument for why kids 3 and up, or any kid who's school age, shouldn't be being vaccinated this summer. So, regulatory affairs is not, I teach at MIT, and the students think that drug approval is the result of a programmed algorithm. And they're like, and they're brilliant, they're all engineers. And when I tell them, no, there's a bunch of people that sort of look like us sitting across the table, and we're trying to, and they work for the FDA, and I work for the drug company, and I've got my argument, and they've got their argument, and we all want to advance healthcare in America, and we have to figure this out together with a limited amount of data. And they're like, you're kidding me. Isn't there a math model that tells you whether a drug is safe and effective? Well, no, there isn't. So it's human judgment.
I think we have time for one more. Yeah.
You are good at keeping trains running. The vaccine train is off the track. If you were Fauci, you really get back on the rail.
Well, I think that trying to remind people that, first of all, pointing out that, let me hear what your concerns are, which, people are doing. Let's listen to people because there's a lot of scary things going on. It's a new vaccine. It's a bad disease. You get bombarded on the internet by all sorts of, crazy false claims. But, if that's what you watch for news, we all create our own reality. So I think, at this point, it's community by community. I think you have to go into community and you have to say, hey, you guys have gotten polio vaccines. You guys get, flu vaccines. You guys get measles vaccines. Over 1 billion people have gotten one or another of these vaccines. And yes, we've had some issues because no drug, they're not miracles, they're toxins. But on average, these are very safe vaccines. And look at what's happening. Look at New England. I mean, look at us. We're sitting here, we're not wearing masks, and we're going to have a good summer. We don't know what's going to happen next. But, I look at the numbers every day. So I think I think it's a community, it's not a scientific effort anymore. The data are there, but it's a sort of a translational, social, cultural. Every community you need a strategy that's going to work from town to town, I have a place in Western Maine, and we drive through Mexico, pretty conservative community. But as many of you may know, Oxford County had a terrible early outbreak of COVID. And, up until like 2 weeks ago, if I drove through Mexico, everybody on the street was wearing a mask. Everybody standing in front of the ice cream parlor, they were all wearing masks. So they figured it out, politics aside, they figured it out. This is not a good thing. We don't want to get it. And I think, so you got to sort of take it town by town, community by community. And I think that they're trying to do that. It's hard. It's very frustrating. I look at the Times every day and I'm like, I can't believe these people don't want to get vaccinated.
All right. Please join me in thanking Bert. Thank you so much, Bert.
Oh. Perfect. Is that okay?
Bert and Will, thank you again for kicking us off this season. Next Tuesday, we'll meet again here. We have COA alumnus Michael Diaz Griffiths, who is the executive director of the Sir John Soanes Museum Foundation. And he'll be here in discussion with Beth Gardner, who is our incoming board chairwoman, which is really exciting. So that will be here tomorrow at 9:30. We'll do these every Tuesday. What did I say? That's right. Exactly. Yeah. You can come back tomorrow if you like. No problem. You're welcome to walk around the building and check it out. If you're not members of the Champlain Society, I'd love for you to join, because on the 31st, we're having our large kickoff party here on July 31st, and that'll be a lot of fun. But thank you all for coming. Thanks to everyone who watched over online in the live stream. Thanks to the crew, and we'll see you next week, if not sooner. Thanks, guys. Yeah.